Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker-Anthony, Scientific Editor of the Molecular Therapy family of journals. In honor of Molecular Therapy's 25th anniversary, this episode is the third in a series hosted by Dr. Joseph Glorioso, Editor-in-Chief of Molecular Therapy, that will cover comprehensive reviews of critical developments in the field of gene and cell therapy over the past 25 years. In this episode, Dr. Glorioso will discuss the reviews, current trends in gene therapy to treat inherited disorders of the brain with author Beverly Davidson, the Children's Hospital of Philadelphia, and The Road Toward AAV-mediated Gene Therapy of Duchenne Muscular Dystrophy with author Jeff Chamberlain, University of Washington School of Medicine. If you enjoy this deep dive into research that is critical to the field, check out two more ASGCT events happening this fall: Advancing Cell and Gene Therapies for Cancer and Breakthroughs in Targeted In Vivo Gene Editing. During these events, in October and November, respectively, you'll be able to hear directly from researchers about their own work related to these topics, or you can submit your own abstract for possible presentation. Visit asgct.org/advancing or asgct.org/breakthroughs for more information. We hope to see you there. And now, Drs. Glorioso, Davidson, and Chamberlain.
It's a real pleasure to have Bev Davidson and Jeff Chamberlain here today. These two guys are real experts in the field of gene therapy. They've been around for a long time. And both these guys were at the University of Michigan when I was there, actually. They've led the fields in treating muscular dystrophy and brain diseases and rare diseases. So I'm going to start out with Bev, who's an expert in delivery to the brain.
Thanks, Joe. It's a real pleasure to be here today and visit with you and Jeff. And I do remember when I was at the University of Michigan, I was a graduate student, Jeff was a new faculty member, and you were a more established faculty member. And it's been a journey.
Yeah.
I mean, starting at the very beginning, I think you recall we were working on brain gene delivery.
Yep.
In early days with viruses from your arena and based on the herpes virus, as well as old school retroviruses, and just kind of ran into a few problems. And then we transitioned to this new kid on the block known as adenoviruses. And they were really great for getting reasonable sized pieces of DNA into cells in the brain, but they really created a lot of toxicity that, really made us look far and wide for better vector systems. And, I think over the last 25 years, the field has really been focused on identifying new adeno-associated viruses, which are similar to adenoviruses in some respect. They're DNA viruses, they're encapsulated, but AAVs don't carry a lot of the viral baggage and hence immune responses into the brain along with your desired cargo. And so our group was among the first to begin to collaborate with Jay Kairini and Rob Cotton to test new vector systems in the brain. And that was a fun collaboration. And we discovered AAV5 was great for the brain. That's now in the clinic in human trials for Huntington's disease. And then tried to advance those for some other disorders. And it really became apparent we needed to think about alternatives to some of these standard capsids. And the field has really kind of marched through a number of different natural serotypes, and more recently, folks have used, advanced learning models and other engineering approaches to create diverse libraries and then identify those capsids that get the viruses to the places they need to be at doses that aren't staggeringly high. So that's, where the virus field has really advanced. And in parallel to that, we've thought a lot about how to design better cargo that go into our viruses. And for the brain, sometimes when you're trying to do a replacement gene therapy, too much replacement is just as bad as not enough. And so many of the investigators in this space really think critically about not only driving the virus vector to the right cells in the right regions, but also expressing those therapeutic products in the right levels. And as we moved into the realm of gene editing, from base editing and prime editing as examples, I think one of the critical questions is, do we need to express those CRISPR enzymes or those other editing enzymes forever? And that's one of the beauties of AAV, is it lasts forever, and maybe we need regulated systems. And so that's been another place where there's been some forays into the developing of either drug-inducible systems or cell-inducible systems to control the gene expression of those cargoes. And then our lab and other labs have worked hard to engineer better ways to knock down dominantly inherited toxic gain-of-function disease genes. And in addition to editing what's now old school, but it's not that old, it's only 25 years old, vectorized RNAi has really moved into the forefront. So I see where delivery to the brain is going to expand in the future, is going to extend beyond viruses into non-viral particles, lipid-based nanoparticles. Right now, some of those systems work great in model systems and maybe even mice. And I'm really, really looking forward to how those new systems evolve down the road. Really just expanding our armamentarium.
One of the things you mentioned in your review was the use of microRNAs to regulate gene expression, and even in systems where you're doing editing. Could you expand on that a little bit? What's your thoughts on?
We were really inspired by the work that came out of the RNA interference field, Andy Fire and Craig Mello and others. Just beautiful work showing the natural process of RNA interference. And my lab and other labs like Marquet's lab, worked hard to build newer expression systems that would allow for more appropriate expression of inhibitory RNAs. And the way that we tackled that problem was to take naturally occurring microRNAs and then engineer them to deliver the sRNA to our gene target of interest. And there are several ways to express those in vector-based systems using, standard promoter systems, even tissue-specific promoter systems. And in some cases, there are non-long coding RNAs that are not microRNAs. They're just long non-coding RNAs themselves that have biological function and can repress either splicing or gene expression. And those have been implemented in various strategies to control gene expression in ways that are really quite novel. And it's been fun.
Yeah, that's fantastic. So I guess one of the things that's I was curious about is people have made all these different capsid variants and so on, shuffling them and looking at naturally occurring viruses. I was wondering, are there new receptors that are being targeted, or really is this an issue of messenger RNA production in the nucleus?
I think it's a bit of both, Joe, and it's a really great question. So the capsids, some of the capsids that have been engineered, if you go back to 2009, when we designed things to target particular receptors and then clone those into the AAV capsid, we could show that we could redirect AAVs from the blood to the brain. And more recently, that's been shown using newer methodologies to advance the integration of receptor ligand sequences, if you will, partial sequences into the body of the capsid. We've taken a very unbiased approach where we start with the aim of hitting the right region and the right cells and put a library into the brain and ask the simple question, where's the needle in the haystack in that library of AAVs, and identify that. And from there, what we're finding is it's probably not a new receptor we've identified. It probably has to do with improved trafficking and expression of those cargo in the nucleus. And that's really what's probably conveying the incredible potency of these newer engineered vectors.
So I guess there's an epigenetic function involved in expression, right? So in some cells, the epigenetics shuts it down, and other cells it does not. But you're right, it has to be that there's some specificity where the virus goes, unless all the specificity is due to gene expression. I don't know about that.
Mm-hmm. Some AAVs go pretty much everywhere. It's, some serotypes just really like to go everywhere, and they persist, for quite some time when you introduce them into the blood. And other AAVs, are taken up quite fast through, adhesion type methods as well as, receptor mediated endocytosis. And I would say that with regards to restricting expression, some of these newer capsids that have been engineered are incredibly specific for certain cell types, even when put into the milieu of a brain where you have this myriad of different neurons and glia and oligodendrocytes and cells that make up your vascular system. And yet these viruses placed in that milieu will go to exactly the right cells when you put them through the proper selection process. So that's one way to control where they go. Another is, as you mentioned, using promoters, and some beautiful work that's coming out in the field now too also uses really specific enhancers. And that's, the beauty in the brain is there are, many different classes of neurons, and it's highly likely that you may want one enhancer for one type of therapy and another enhancer for another type of therapy. And so it may be getting to the point where it's somewhat of a web-based design tool to get your vector of choice for your disease of choice.
Are you using AI tools at all for, you've talked a lot about gene editing to line up the right RNAs and all that kind of stuff, guide RNAs?
Most of the tools for the designing of guide RNA cargo are pretty simplistic, almost like a BLAST type program. And there's your tools on various websites where you purchase the tools to, identify the best in class compounds to end up adding into a vector-based system, like language learning models and other assisted intelligence designs. I think, some of that's being used in the capsid library format, but a lot of us are letting the biology of the animal do the work for us, and really it's setting up, selection paradigms where the cream really rises to the top after just a couple animals, and you have your winners.
Yeah. How good is that a selection and say mice going to primates to going to humans? Is that
Well, that's been a bit of a challenge, and at least in our laboratory, we've taken a different approach. We start with non-human primates, and so far the work that we've done to identify, brain capsids for, disease X versus disease Y, they have very different characteristics, but they still tend to work beautifully in mice systems, strain independent, as well as human neurons and various classes of neurons. So probably going down is better than, starting with a lesser species and trying to make it work up.
I see. So one of the things about brain gene therapy is not just delivery, which is a big deal, but also the complexity. You have multiple genes involved and all that. And what's your view about complicated diseases like Parkinson's? Can we approach that with single gene changes, or do we have to hit multiple sites? What's your thoughts about that?
Yeah, I mean, I don't work in the field of Parkinson's for a reason. too close.
It's
So
too hard.
yeah, but I mean, we have the right vectors now to get to the areas of the brain, using relatively low doses that make scaling for the large number of patients that have Parkinson's disease tractable. And there are familial forms of Parkinson's disease where the genetics is known, the biology is relatively straightforward, and so the approaches could somewhat work. Now, creating a one-size-fits-all therapy for disorders like ALS or Parkinson's disease is a bit more nuanced until that is where I think some advanced learning model systems will really help us identify better target therapies, as well as some way to understand if our therapies are working through advanced, biomarker discovery.
That's great. So I want to ask you just two more questions.
Mhm.
First of all, Huntington's disease is a terrible problem, and you have these CAG repeats and all that. So, uh, do you think gene editing can solve that problem?
Well, I mean, that's a great question, Joe. There's a lot of genes that have CAG repeats in
Right,
in
right, right.
our genome, and I think one of the things that we've learned, over the years is that gene editing is not as easy as it sounds. A lot of the machinery is quite big and doesn't fit well into an AAV system, although newer and better reduced size editing systems are coming online, which is incredibly exciting for the field. Then you still have that question of, do we want expression forever?
Yeah.
I think editing the CAG repeat, there's several ways to go about that. You probably don't want to just get rid of that, kill that gene product, because we know that knockout of Huntington is complete knockout of Huntington is problematic. The cells need Huntington. So, many of us are going at this in different ways, and that is to target disease modifier genes that actually exacerbate disease progression and age of onset. And so, really trying to convert an aggressive HD phenotype to a mild HD phenotype based on what we know about the genetics of HD is where a lot of folks are going now, as opposed to knocking out the CAG repeat.
I see. Well, that's very interesting. So, one last question. So, what do you think in the brain is going to be a great target in the future? I know that ALS and so on have been, that's not in the brain, but people are working on that. But I think, I read just the other day that the amount of people at about 2040 that have AD is going to be like 20 million or something insane like
Yeah.
that. So I was just wondering what you guys are targeting at the moment. Where's this going?
Yeah, I mean, in my group, which is really all I can speak, confidently about. I can't say what everybody else is doing, but we still are targeting, these rare disorders that affect children, lysosomal storage diseases, and we are still advancing our work in the Huntington's disease arena. Many of the vectors that we've engineered are going to find application to disorders like ALS and Parkinson's disease. And really, that's through the secretion of soluble proteins that may be protective. So, take, for example, some of the antibody-based therapies that are delivered to patients at grams per dose repeatedly throughout a patient's life. There are ways to express reasonable amounts of protein in the brain through these engineered vector systems that may have
Yes.
applications to a disorder like Huntington's disease. In effect, bringing the cost of those medications down tremendously, because these newer engineered vectors are no longer in the order of hundreds of thousands of dollars per dose per patient. Because they're so potent and efficient, you're 20,000 per dose per patient, which makes treating these disorders feasible, for which there are millions of individuals in the planet.
Yeah, yeah. Let me turn to you, Jeff. So I really enjoyed reading your review as well, and
Yeah.
I think, you've been involved in Duchenne muscular dystrophy forever, right? And
Seems like it. Yeah.
30 years maybe or something. And people have worked on this and so hard over the years. And you've made real progress recently with AV and using mini dystrophin or micro dystrophin or whatever, but also your recent work with inteins and mRNA splicing and that kind of thing, which is that's very interesting stuff. So maybe you could tell us a little bit more about the history of this, which you outlined beautifully, and then where you think it's going to go from here.
Sure, I'd be happy to. Well, I can't help but think back as Beth Davidson commented to the early days at the University of Michigan. It was such an exciting period. It was really the hotbed of gene therapy back then.
Yep.
And there were so many good labs. I mean, we were all interacting even with Francis Collins back then, who was
Yeah, that's right.
nucleating force for all of us. Before leaving for NIH, of course. And I thought one of the highlights of a recent ASGCT annual meeting was when Bev Davidson, when she was serving as president, was able to get Francis Collins to come out and give a talk about his views on gene therapy and the Genome Project and all that. That really brought back a lot of memories. One of the other exciting things I think in the early days at Michigan was in many ways, we didn't really know what we were doing. Everybody wanted to do gene therapy. We knew what the targets were. We knew many of the challenges, but trying to figure out how to overcome those was a huge issue. There was also a lot of encouragement or pressure from the institution at the time to try to develop intellectual property and patent things. If I recall, Joe, I think you may have written the first patent on gene therapy.
I
Whatever
don't know
issue
about
that
that.
patent was maybe a little ahead of your time because I think it expired a long time ago. And
Yeah.
it's kind of ironic thinking back. So much of the original intellectual property around gene therapy, those patents expired well before any commercial application of the
Exactly.
technology. So that's it's kind of funny to think back to to those early days.
That's
In
right.
any case,
We had
I
people like Craig Thompson there, Jim Wilson. It was amazing. Bill
that's
Kelly.
right. It was a hotbed. Where everybody has since become dispersed. That's sort of the nature of our field. You go where the opportunities are.
Yep.
And it's actually in terms of the review article that my group contributed to your issue here. It was interesting going back thinking about the early days and how many of the people that were major contributors back then really kind of moved on to other fields in other things and unfortunately, a number of them have retired since then. So somehow
Or died.
or died. Yeah, I'm still hanging in there. We'll see how long that lasts, but hopefully we'll be able to keep going for a while. But part of it, was the challenges. It's obviously my group has always been interested in the muscular dystrophies, and it's presented its own unique set of challenges, like all gene therapy targets do. In particular, with the musculature being 40% of the human body, it was always a daunting issue of how we could possibly deliver a gene body wide. Also, in the case of Duchenne muscular dystrophy, that we were particularly focused on, the Duchenne gene has multiple promoters that are expressed in different tissues. It's an alternatively spliced transcript, and we really weren't sure what we would need to deliver and where we would need to deliver it to. And on top of that, the dystrophin gene was soon realized to be the largest known gene. It's over 2 million base pairs in size. So, just trying to handle a gene that large was a challenge in itself. So, many of the people in my group used to come to me and say, this isn't possible. Why are we doing this? What is your vision to get a therapy developed? And basically my reply was, I have no idea how to do this, but let's just take it one day at a time. And, we'll ask very focused questions, try to solve problems that come up, and hopefully we'll make progress every year and keep going. And that worked out to be a reasonably good strategy. We're still not there yet. There's a lot of progress that needs to be made. But what's exciting is that some of the earlier work we did in terms of vector development and creating smaller dystrophins and microdystrophins has now led to approval of the first gene therapy for a muscular dystrophy. The FDA approved a microdystrophin vector just last year. It's not perfect. It's an important breakthrough, and now the question is how can we improve upon that? But early on, a lot of the questions we were asking were focused on the dystrophin protein itself. It's so big, we didn't know what it did, so we started trying to break it down into different pieces and ask what are the different functional domains, which ones are important, which ones are maybe less important. And critical to that was, an intimate knowledge of the patient literature, knowing, clinical data related to specific mutations and the effects that they had. And in particular, the Duchenne muscular dystrophy and the milder version of it, Becker muscular dystrophy, are somewhat unique in many ways in that a lot of patients have very large intragenic deletions that remove parts of the dystrophin protein. And yet, in many cases, that leads to a fairly mild phenotype where you have individuals that can remain walking in rare cases for their entire life, whereas other mutations can lead to very severe versions of the disease that result in early lethality. So trying to sort that out would play an important role in trying to figure out what we needed to deliver to different tissues in the body. Then we also focused a lot, as you were alluding to earlier in your discussions with Bev, on how to regulate production of the protein. You don't want too little, you don't want too much. We were able to gather some of that information again from the clinical literature, but we also addressed that directly in mouse models for Duchenne muscular dystrophy. And realized that, again, we were in a pretty good position with Duchenne muscular dystrophy. We were able to, in some cases, generate animals that were making 50 times normal levels of this dystrophin protein, and we were not able to find any adverse consequences of that. We're also able to realize that if we could achieve uniform delivery of dystrophin to muscles, which was a bit of a challenge, you really didn't need all that much either, maybe only about 1/5 of the normal amount. So it gave us a huge range of levels that we thought might be therapeutic without inducing other problems. But we still needed to figure out ways to produce those levels in all the different types of muscles in the body. There's almost 600 different muscles in the body. Many of them have very unique transcriptional profiles. But we were very fortunate to kind of take advantage of work that I had been doing as a graduate student with Steve Hauschka, who's at the University of Washington, where I'm back there now. Steve's lab had taken a long interest in understanding muscle-specific gene transcription, and in fact, when I was in his lab, I cloned one of the first muscle genes, the muscle-creatine kinase gene, and we identified the first muscle-specific enhancer element. And, I thought, that's great. I'll leave that behind. Steve continued working on it for many years, but once I got involved in gene therapy, we realized we needed muscle-specific promoters. And Steve and I teamed up again and are still collaborating, 40 years later on finding optimal transcriptional regulatory elements to drive gene expression in muscle. And this dovetailed nicely with the various studies of structure function of dystrophin and what are the optimal versions of the protein to deliver. Delivery became a huge issue. Together with Bev and many others at Michigan, we were desperate to try any kind of delivery system we could get our hands on. There was some early interest in direct plasmid injection into muscle. Then we started working with some retroviral vectors and then moved on to adenoviral vectors. And as Bev noted, adenovirus was the thing for many years. You could grow it up into large quantities. You could inject it into a lot of different tissues, get very robust levels of gene expression, but it just wasn't persistent. There was still a little too much toxicity or immune response issues with adenovirus. And in the case of muscle, we were never able to figure out a way to do widespread delivery. We could get fantastic gene delivery and expression by intramuscular injection, but that doesn't do you a lot of good for all the muscles throughout the body. So, as a lot of people say, along came AAV, and it seemed to be a miracle, except for this minor issue that AAV will only hold about 4.5 kb of DNA, and we were dealing with a 2 million base pair gene. So, minor obstacle there. So, but it did dovetail with our structure function studies and with Steve Hauschka's work on miniaturizing muscle regulatory cassettes. And all that came together, and we figured out a way to make a functional microdystrophin, as we named them at the time, with a very small but powerful muscle-specific expression cassette. We were able to fit that into AAV. And that was really the beginning of these AAV microdystrophin gene therapies. The problem at the time still was we could only do intramuscular injection. We weren't sure how to do widespread infusion. And around that time, I moved my lab out to the University of Washington to collaborate with a lot of people here. We were recruited out here by George Stamanopoulos, who of course founded the American Society for Gene and Cell Therapy. And there was a great group of people out here interested in gene therapy that gave us a lot of support. And in particular, we started working with with a couple of labs that were really good at making AAV vectors, and we were able to ramp up our production capabilities to the point where we could inject intravascularly amounts of vector that, even a year or two earlier, we never even dreamed of being able to make that much. And we realized that if we could achieve a certain threshold dose of AAV into the vasculature, it could be extravasated out of capillaries and target muscles throughout the body. So that, to us, that was a miracle, something that we maybe never thought would happen. And so we had a lot of pieces in place. We were able to deliver genes body-wide to muscle, express them in a muscle-specific manner, and produce miniaturized functional dystrophins. And again, that's what led directly to the recent FDA approval of gene therapy for muscular dystrophy. Now, unfortunately, it's not perfect. We're only able to deliver proteins that are only about 30% the size of normal dystrophin. There are issues in getting enough dystrophin into human muscle compared to animal models. And this gets back to your questions earlier about delivery systems. Where do you select? Do you start with mice? Do you start with non-human primates and all that? And one of the things we've seen is that, we can essentially cure muscular dystrophy in the mouse, but doing the exact same thing in the clinic is only partially effective. It's not there. So you've got to really focus on the ultimate target, the patient population, and work from that. So we're taking a lot of the clinical data that's coming out from a variety of biotech companies now, and now trying to work backwards and figure out how can we re-engineer these systems and make them work better. And that involves trying to take advantage of new AAV vectors that a lot of laboratories are developing, new and improved muscle regulatory cassettes with better enhancer elements. And then ways to make larger, more potent dystrophins, and we've tried a lot of different ways to make bigger dystrophins through trans-splicing and homologous recombination. But the method that is really working the best for us, I think now, is using these split inteins that you mentioned a minute ago. Split inteins are really a kind of an amazing system where you can take very small peptides and combine them onto two different pieces of a protein. And if they're expressed together in the cell type, they come together, and it's an incredibly fast and efficient reaction that merges the two proteins you're trying to join and excises the split intein in an incredibly rapid and efficient manner. And the result of that is we can deliver 2 or 3 different AAV vectors simultaneously. They're taken up together simultaneously by muscle cells throughout the body, and they're able to make significantly larger dystrophins. And two things came out of that were not entirely anticipated. One is that we're actually achieving higher levels of gene expression with these dual and triple vectors than we were with the single original microdystrophin vectors. And I think the reason for that is that these larger proteins are more natural type proteins. They're more stable, and they accumulate to higher levels. So that was kind of an unexpected bonus. The other thing that was partially expected was that these larger dystrophins, they're more functional, they're more potent, and we see a more significant ability to not just prevent the development of muscular dystrophy, but to achieve a reversal and a phenotypic improvement, even in older dystrophic mice. The big question now is, will this translate back into the clinic, and can it be done safely, efficiently, and will it have a better impact on the disease? So we'll see how that goes. Still got a lot of work to do, and we're excited to keep going.
Fantastic story. So, one of the things you mentioned in your review was the activation of utrophin,
Yes.
and is that another solution? Just because you don't have the immune problem that you have with putting dystrophin in the body for the first time.
Exactly. Yes, we have a whole parallel program going on to try to deliver utrophin vectors rather than just dystrophin vectors. And that goes back to the very early days of gene therapy, even almost pre-gene therapy. I remember a lot of people saying that a lot of the early targets of gene therapy were really genetic diseases because they're a little more defined and perhaps more accessible to try to treat. But there was always this worry that with a genetic null allele disease, where you're not making the missing protein, that if you're successful at gene therapy and can deliver that protein, it may be viewed as a foreign antigen and you may elicit an immune response and then get rejection of the tissues that you've treated. Fortunately, that has not been seen in the clinic as much as I think we were originally worried about, but it is being seen in a certain extent, and in particular, it's an issue with Duchenne muscular dystrophy because many of the patient mutations, almost 2/3 of them, are partial gene deletions, so that there are parts of the gene that are absolutely cannot be expressed, that the immune system is going to be naive to any epitopes that are in that protein. So what's been remarkable is that very few patients treated with gene therapy so far with Duchenne have actually elicited an immune response. The vast majority haven't, but about 1% of the patient population has in fact mounted an immune response against dystrophin. They have indeed then rejected the vector, and that's led to some very serious adverse events. So what do you do about that? The current approach in the clinic, unfortunately, is simply to exclude patients that have deletions of portions of the gene that appear to have potent epitopes in the dystrophin protein. And so we're taking a couple approaches to try to address that so that we can, come back and be able to enroll all patients in the clinical trials. And one way to do that is to deliver this utrophin protein. So utrophin and dystrophin, of course, are very similar proteins. They appear to have arisen by gene duplication. Utrophin has been shown through the work of Kate Davies and others to substitute reasonably well for dystrophin if you can overexpress it in the right places. It's not quite as potent as dystrophin, but it works pretty darn well. So we published a paper many years ago with a so-called micro-utrophin instead of a microdystrophin. We're taking advantage of the split intine technology to deliver full-length utrophins as well. And that may be one way to overcome this. The other approach we're taking is actually to go in and identify specific epitopes that have been identified in the clinical trials. And again, we're dealing with an enormous protein, but surprisingly, very few epitopes have really come up in these clinical trials that have led to adverse events. So with an ability to identify those epitopes, we're actually trying to take advantage of artificial intelligence programs to sort of model the shape and the structure of these epitopes, how they interact with MHC receptors and things of that nature, and then again use AI to redesign the epitope to make very minimal changes to the protein structure to preserve functionality, but now come up with a protein that may not be able to elicit an immune response. It's going to be a little harder to test that strategy, I think, in the clinic. We've got some encouraging data in animal models, but it gives us sort of two different approaches to at least develop vectors for some of the patients that are currently being excluded from clinical trials. And which also leads to the possibility that you may end up with multiple vector systems that you will apply to different patients depending on their specific mutations.
Right, right. One of the issues, I guess, is the fact that dystrophin has a variety of mutations, so it makes gene editing more difficult, I guess. So I was going to ask you too, the mini dystrophins do they lack domains of dystrophin that are really required to get, full complementation?
Well, it's a complicated question. On the one hand, the success that we've had in developing microdystrophin sort of shows that you can make a protein only 30% of the full-length protein and still have it work reasonably well. But you take a hit. There are certain functional deficiencies in those smaller proteins, and I think I believe in Mother Nature and evolution and all these things. And I think there's a reason the dystrophin gene and the protein are as large as they are. I can't quite figure out what that reason is, but we know that you go back through evolution, all the way back to chickens or even sea urchins. The dystrophin gene is enormous in every species that has been looked
Really? Oh,
the protein is enormous in all of these different species. So there's presumably been some selection for that. So, ultimately you want to be able to deliver the full length dystrophin protein back into cells. We'll take what we can get. If we can come up with a reasonably effective therapy, leaving out some of those domains, then so be it. But our ultimate goal is to be able to deliver the full length intact protein, but this does relate to gene editing. I mean, in many ways, gene editing is a very attractive approach for the muscular dystrophies. If you can induce mutations, particularly a lot of the targets are splice site mutations to skip over exons that might have a mutation in them, then you can make, not full-length dystrophins, but perhaps dystrophins that are only missing very small functional domains. And some of those larger proteins are going to be quite potent. Not all of them, though. You run into these issues where even if you can produce a slightly smaller dystrophin that's only missing a small functional domain, sometimes those proteins are not all that stable, and you can run into issues in how much of the protein you can produce. So you got to balance not only the functionality of different domains, but the stability of the proteins that are lacking those domains. The other issues you mentioned, Duchenne muscular dystrophy is a new mutation syndrome. It had the dystrophin gene has one of the highest known rates of spontaneous mutation of any human genetic locus. So there are tens of thousands of different mutations out there in the population. Fortunately, they tend to fall into clusters or classes of mutations that affect certain parts of the gene. But I think it's estimates out there are that if you wanted to have an effective gene editing strategy that would be applicable to, let's say, even 80% of the patient population, you would probably need 20 different individual vectors using different guide RNAs and things like that to target those. In many cases, that may be the best way to go because you might make slightly smaller dystrophins, but pretty big ones. In other cases, you've got mutations of critical functional domains where you're going to have some problems. And then again, a number of the deletion mutations in patients really take out very large parts of the protein. So, I hear a lot of people talk about, well, the gene editing, we're going to make almost a full-length dystrophin. Well, that's only in a subset of patients. There's many other patients where your editing is only going to make a very, very small protein. So, but the other advantage is you can potentially get permanent correction, particularly if you can get gene editing into the muscle stem cells. You may have a lifelong therapy, which again is the holy grail in the field there. The problem with that though is these AAV vectors are really bad at getting into stem cells in general. And I think there's some hope out there with a lot of the engineered AAVs, the new designs that are coming out that like Bev was alluding to earlier. So I think we're getting to the point where we may be able to start targeting muscle stem cells much more efficiently, and that's going to be huge for the field.
right. Well, that's very exciting. I think it's amazing all the work that's been done to develop these treatments. Had one last question. There was a recent death, I guess, in a subject that was treated, and Terry. I was talking to Terry Flout recently. He was saying that they had a lung disease rather than, a heart problem. So they had, respiratory distress syndrome. And I wondered if you'd ever seen AAV go into the lungs like that.
Well, it depends on the serotype a little bit, but one of the things you run into, I think the patient that you're referring to was one of the oldest patients that's
Older
been
patient, yes.
in a Duchenne gene therapy trial, and one of the things that may have contributed to that is that these older patients have a tremendous loss of muscle mass.
Mm,
And so I think rather a lot of the normal target of the
they
AAV
weren't there,
trying to
yeah.
get into muscle weren't really there anymore.
Yep.
And for reasons that are still a little unclear, the vectors seem to be taken up by a lot of lung cells and perhaps a lot of inflammatory cells as well. Certainly a lot of cardiac muscle. And there have been some instances of cardiomyocytis in some of these Duchenne trials that have led to some severe adverse events. So, exactly what went on is a little unclear, but your description is right on. It appears to be, certainly a partially a systemic inflammatory syndrome issue with and targeting lung inflammation leading to rapid respiratory failure. Unfortunately, I think in so far in the Duchenne muscular dystrophy field, there's been about 900 patients treated with AAV vectors so
It's a
far.
rare event that this is.
There's, I think, 3 or 4 deaths. Unfortunately, there was another one just last month in a Sarepta trial
Right.
where the patient developed a severe liver injury. And, it's unclear what happened there. Again, that's a rare event. You often get, minor, liver issues that can lead to elevated transaminases. That's usually transient. In this case, it unfortunately spun out of control. There's some suggestions about reactivation of cytomegalovirus or things like that. On the other hand, reactivation of some of those viruses has been seen in a number of other patients without adverse events. So it's unclear what's going on. There's a lot of interest in the muscular dystrophy field and trying to identify is there something unique about the patients that have developed very severe adverse events
Right.
that's makes them stand out. Are there modifier loci, rare genetic polymorphisms? Very unclear. I think we need to sort that out to try to be able to predict which patients may be more likely to develop a serious adverse event.
Is there any connection with MHC types? Or?
Possibly, it's still being worked out. So there's a large consortium between a lot of the biotech companies working in this area and many of the researchers studying it to try to develop more information on various patients. Starting to move in the direction of doing whole genome sequencing for a lot of the patients that have been in these trials to see if any subtle things stand out. If it could be with MHC types and all that. There's some hints that may contribute a little bit, but I don't think that's the only answer, the only reason so far.
Bev, last question for you. Okay. Is the brain a privileged site immunologically?
Well, I think the brain and the eye seem to result in a little less robust immune response, but that can certainly be blown apart by dose. So, as you use very high doses of vectors, regardless of that route of delivery, you can create a number of problems in the brain. I think we were all encouraged with the original eye gene therapy trials where they treated one eye and then, were asked by regulatory agencies to, before advancing to treating both eyes, to go back to those same patients and treat the second eye. Many of us were worried that that was going to reduce the positive impact on those initial treated eyes and allow for nothing in the second treated eye, but it turned out not to be the case. There are situations where direct parenchymal injections into the brain have been done really safely with and repeated, so I think if we continue to make strides in creating highly potent AAVs where the doses delivered are orders of magnitude lower, I think it becomes very attractive to both be safe without creating any immune response in the brain, but also even opens up the possibility for redosing. So
Yeah. All right, guys, I think we have to close this. You've been so wonderful to stay with me for almost an hour, right? So I really appreciate that, and so do our viewers or our listeners, I guess. And I'm sorry I'm gonna miss you in New Orleans, but
yeah,
anyway, it's a special situation, right?
yes,
So
very special.
Jeff, you're the outgoing boss, so I look forward to seeing you there. And so again, thank you so much for all your help and your great reviews.
Sure. It's going to be a
Thank
great
you,
meeting this
Joe.
year. New Orleans is always a fun place to visit. So.
Yeah.
Yes.
Thank you, Joe, for your leadership and for over the years, not just, with taking on the helm as editor of molecular therapy, but also for all the hours, many, many hours you've you've spent in this space also.
Yep.
So always great to see my buddy Jeff.
Yeah. Great
Okay.
seeing both of you too. Yeah.
All right. Thank you so much.